Today, let us have a discussion on DNA denaturation and renaturation. Introduction. All living organisms possess genetic instructions that determine growth, development, and reproduction. In the living organisms, two chemically similar molecules of DNA and RNA are responsible for carrying and the expression of genetic information. Except some virus, genetic instructions of the living organisms are encoded in the molecule called deoxyribonucleic acid, popularly known as DNA. It is basically a long molecule that contains coded instructions for the cells. For a long time, attentions of scientists were directed at understanding the structure of DNA, particularly the sequence. However, the knowledge of the structure of DNA is not sufficient to understand biological processes. Understanding how structure of DNA affects the equilibrium properties and the dynamics of DNA is also important. In fundamental genetic processes such as transcription and the replication, DNA undergoes dynamic change with high complexity. In this lecture, basic concepts regarding DNA denaturation and renaturation is highlighted. Before presenting these concepts, it would be necessary to recall the double helical structure of the DNA. Double helical structure. Deoxyribonucleic acid is a polymer formed by several nucleotide monomers. Each polymer consists of backbone of deoxyribose sugar molecules linked together by phosphate bonds. Each sugar molecule is covalently linked to one of the four bases, namely adenine, guanine, cytosine, and thymine. In most of the organisms, DNA consists of two associated polynucleotide strands that wind together to form a double helical structure. Only in some viruses, single-stranded DNA molecule exists. Double helical structure was proposed by James D. Watson and Francis I. C. Crick in 1953 based on analysis of X-ray diffraction patterns along with careful model building. The Watson and the Crick model of DNA proposed that two right-handed polynucleotide chains are coiled in a helical fashion around the same axis, thus forming a double helix. The orientation of the two strands is different, that is, their 5' to 3' directions are opposite. We call this arrangement as antiparallel. In the double helical structure, adenine in one strand is paired with thymine in opposite strand through two hydrogen bonds. Guanine is paired with cytosine through 3 hydrogen bonds. The association between a larger purine and a smaller pyrimidine are usually called Watson-Crick base pairs. Two polynucleotide strands in which all the nucleotides form such base pairs are said to be complementary. The two strands are held together by formation of hydrogen bonds between base pairs on the opposite strands. Though these bonds are weak, presence of large number along the entire length of the DNA make them a significant force. Apart from hydrogen bonds, hydrophobic interactions, van der Waals force also contributed to the stability of the DNA molecule. Sometimes, two strands of the DNA may separate into separate strands in a process called denaturation. Denaturation. In simple terms, denaturation is a process where nucleic acid or proteins lose the quaternary structure, tertiary structure, and the secondary structure by application of some external stress or compound. DNA denaturation is a process where double-stranded DNA unwinds and separates into two single strands through the breakage of hydrogen bonds. It also involves disruption of the hydrophobic interactions of the bases. So, denaturation can be described as a process of formation of single-stranded DNA from double-stranded helical DNA. DNA denaturation may be induced by high temperature or applying some chemicals such as urea and the formamide. Biologically induced denaturation is observed during DNA replication and transcription. Thermal denaturation of DNA is often termed as melting. Thermal denaturation is the most common type of denaturation. In temperature-mediated denaturation, with the increase in thermal energy, the molecular motion also increases. It results in the breaking of hydrogen bonds and the other forces that stabilize the double helix. Then, the strands separate, driven apart by the electrostatic repulsion of the negatively charged deoxyribose phosphate backbone of each strand. Near the denaturation temperature, a small increase in temperature causes a rapid loss of the multiple weak interactions holding the strands together. DNA from different cell types have different melting points. At the physiological pH and ionic strength, the melting temperature of DNA is between 85 degrees centigrade and 95 degrees centigrade. Temperature at which a particular DNA is denatured is characteristic feature of the molecule. It is expressed as melting point of the DNA. The melting point or midpoint temperature Tm is the temperature at which half the DNA is unwound. Above Tm, DNA is single-stranded, while below it DNA remain double-stranded. Melting of double-stranded DNA can be monitored by the absorption of ultraviolet light at 260 nanometer. As regions of double-stranded DNA unpair, the absorption of light at these regions increases. On the other hand, light absorption by single-stranded DNA changes much less as the temperature is increased. The melting temperature depends on base composition, length of the DNA, ionic strength, pH, and denaturing agents. If several samples of DNA are melted, it is found that the Tm is highest for those DNAs that contain the highest proportion of guanine-cytosine pair. There is linearity between the Tm of DNA and GC content in many species. This relationship between Tm and GC content arises due to the fact that guanine and the cytosine form 3 hydrogen bonds in base pairing, whereas adenine and the thymine form only 2. At the same time, GC pairs are more hydrophobic than AT pairs, so they stack better, thus affecting the stability. A DNA that consists entirely of AT base pairs melts at about 70 degrees centigrade, and another DNA that has only GC base pairs melts at over 100 degrees centigrade. As such, percentage of GC base pairs in a DNA sample can be estimated from the Tm. Higher the GC content, higher is the Tm value. The ion concentration also influences the DNA denaturation. It is because negatively charged phosphate groups in the 2 strands are protected by positively charged ions. When the ion concentration is low, this protection is decreased, thus increasing the repulsive forces between the strands and reducing the Tm. When there is low salt concentration, ions that stabilize the negative charge on the 2 strands are removed. It results in increase in repulsive forces. In another case, extreme pH value denatured DNA at low temperature. At low pH value, the bases become protonated and thus positively charged and repel each other. At high pH, ions can pull protons from base pairs, making them negatively charged entities. It makes these bases to repel each other, resulting in separation. Chemicals that can cause denaturation in DNA are called denaturants. They may be competitive denaturants, covalent modification denaturants. In the competitive denaturants, there is competition for forming hydrogen bonds with the electronegative centers of the nitrogenous bases. Such properties are exhibited by urea and the formamide. At high concentration of such denaturants, the competition for hydrogen bonds favor interactions between denaturant and the nitrogenous bases rather than between complementary bases. As a result, the two strands separate. Covalent modification denaturants can modify the electronegative centers of the nitrogenous bases and thereby block the formation of hydrogen bonds between complementary bases. Covalent modification is reversible. Effect of denaturation. When there is denaturation, the native double helix undergoes transition into a randomly looped form, which is a single-stranded or denatured DNA. Along with these structural changes, following changes are observed in the properties of DNA. Number 1, increase in absorption of UV light. Denaturation leads to the abrupt change in the absorption of ultraviolet light. When absorbance of light is measured at 260 nanometer through spectrophotometer, it is observed that absorbance is increased as the DNA becomes denatured. This phenomenon is called hyperchromatic effect or hyperchromacity or hypochromism. It is due to unstacking of base pairs. The rate of increase of absorbance is observed to correlate with the degree of denaturation. Number 2, decrease in specific optical rotation. Double-stranded DNA exhibit a strong positive rotation that highly decreases with denaturation. Number 3, decrease in viscosity. DNA solution exhibit high viscosity because of relatively rigid double helix. Denaturation causes a marked decrease in viscosity. Renaturation. Denaturation of homogeneous DNA is reversible. When heated solutions of DNA are slowly cooled, annealing occurs with return of the coil to helical configuration. This process is called renaturation. In short, renaturation is a process of formation of double-stranded DNA from single-stranded denatured complementary DNA strands. It involves re-annealing or formation of hydrogen bonds between complementary base pairs. When there is lowering of temperature, increase in the ion concentration or neutralization of pH, the two complementary strands may reassociate into a perfect double helix. This process is renaturation. It depends on time, the DNA concentration and the ionic concentration. If the DNA is very long and complex, renaturation may not be precise. Thus, the rate of renaturation kinetics, can give idea about the complexity of a DNA molecule. The process of denaturation and renaturation of DNA are the basis of nucleic acid hybridization technique. This technique is used to study the relatedness of two DNA samples and to detect and isolate specific DNA molecules in a mixture containing numerous different DNA sequences. Application of DNA denaturation. DNA denaturation can be used to detect sequence difference between two different DNA. In this method, DNA sample is heated and denatured into single-stranded state. The mixture of DNA is cooled to permit strands to rehybridize. Hybrid molecules are formed between similar sequences. If there are any differences between those sequences, there will be disruption of the base pairing. DNA denaturation can be used to study the genome size and the complexity as well as relative proportion of single copy and repetitive sequences. Conclusion. Study of DNA denaturation and renaturation have contributed to many aspects of molecular biology. Based on the properties of nucleic acid denaturation, methods such as polymerase chain reaction, southern blot test, northern blot test, and DNA sequencing techniques were created. Nowadays, DNA is used for characterizing and differentiating a species. One popular molecular biology technique is DNA-DNA hybridization that measures the degree of genetic similarity between pools of DNA sequences. This technique is generally used to determine the genetic distance between two organisms and used extensively in phylogeny and taxonomy. DNA-DNA hybridization technique is based on denaturation and renaturation of DNA.